Hi guys, my name is Moesha Morris, and I'm going to be teaching you about Chapter 12, Mitosis. Let's start. Mitosis. What I'm going to be teaching you is mitosis to cell cycle, interphase, two kinds of cells, cell replication, interphase, again, stages of mitosis, more additional information. Okay. What mitosis is? A type of cell division that results in two daughter cells, each having the same number and kind of chromosome as the parent cell nucleus. The cell divides and produces two identical copies. And then mitosis consists of 4 stages. Okay. Cell cycle. A cell cycle is a complex sequence of events by which cells grow and divide. Purpose of the cell cycle is to replace older cells, make new cells, help the body grow, and repair broken bones, and more. This is a chart. This is a drawing of a cell cycle that I drew. First phase is G1 phase, and that has to do with cell growth. Second phase is S phase, DNA replication. So the cells start dividing and multiplying, making more. G2 phase, which is a preparation for mitosis. So this phase prepares the stages to go on to the stages of mitosis. Mitosis, cell division, which has to do with stage 1, prophase, stage 2, metaphase, stage 3, anaphase, stage 4, telophase. Mitosis only has 4 stages, but cytokinesis is something that is added in the information of this chapter too for the cell cycle. All this together is interphase. Okay. Usually I would introduce this with the stages, but I'm going to introduce this first because you need to understand what it has to do with this next I'm going to teach you. Interphase is the resting phase between successive meiotic divisions of a cell, or between the first and second division of meiosis. But we're not talking about meiosis. We're talking about mitosis. A cell spends most of its life in this phase. So, stomatic cell cycle. This is what's a stomatic cell cycle. And how it starts off is interphase. 1 chromatode per chromosome, and this is a chromatode. Then it goes on to interphase and then synthesis. Then it produces 2 chromatodes per chromosome. Then it goes on to interphase again. Then it goes into mitosis. And then mitosis comes on to produce 2 cells. Stomatic cell is any cell of a living organism other than the reproductive cells. Stomatic cells have a diploid number of chromosomes, 2 sets. 2 sets, remember, 2 sets. That's what diploid is. Uses process of mitosis. This is only in mitosis, a stomatic cell in diploid. Involved in and is not involved in sexual reproduction. So stomatic cells has nothing to do with sexual reproduction. It only has to do with other living organisms like your liver, your cardiac, and other stuff. Gamete cell. Gametes are an organism reproductive cells. They are also referred to as sex cells. Female gametes are called ova or egg cells, and male gametes are called sperm cells. Gametes are haploid cells, and each cell carries only 1 copy of each chromosome. Zygotes, which has to do with gametes, is 2 gametes, which is a fertilized egg. So a sperm and an egg together makes 1 fertilized egg, which is a zygote. And this is a picture of a nucleus containing information in the chromosome, which would be an egg. And then this would be a sperm, and that's the head of the sperm, and the nucleus is in there. So this is the egg cell, and then this is the nucleus that's in there, and the nucleus in the egg cell. So this is the sperm, and the tail allows the sperm to swim into the egg, which will make a fertilized egg. That's what gametes are. Cell replication. So this is what cell replication is. How it starts off is 2 separate chromatids. Then it goes on to DNA replication, which makes it multiply for each chromatid. And so this would be like the mothers, and this would be like the fathers. And then the central the spindle fibers will attach to the chromosomes, and then it goes into mitosis, which will separate the spindle fibers, will separate them, so you have a mother and a father, and then it will go into two diploid cells, which is like these two. So you have your mother and your father in each. Replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. So replication is very tightly controlled. Why? Because enzymes carry out the oxidation of sugars in tightly controlled series of reaction. It ensures that each genomic region is replicated fully and only once during each cell cycle. It protects itself. It's to protect itself so it can control the cell numbers and tissue size. Okay, next. Stages of mitosis. Okay, we have the first stage of mitosis is prophase. Well, actually, where's my interface? So all together, this has to do interphase, which you already know, the resulting phase between successive meiotic divisions of a cell, or between the first and second division of meiosis, and the cell spends most of its life in this phase. Stages of mitosis. Prophase. The first stage is the chromosomes appear condensed, and the nuclear envelope is not apparent. Nucleosis disappears. Separation of centrosomes, formation of the meiotic spindle. So they're all together. And as you see, this black part is the nucleus. So, like how they said the nucleus will disappear. And as you can see in metaphase, it's disappearing. So in metaphase, is the chromosomes line up across the center of the cell equator. So this is the cell equator, and this is the spindle fibers, and that's the nucleus disappearing. It's disappeared. Anaphase. The chromosomes have separated and are moving toward the poles spindle fibers. The spindle fibers pull apart centromeres, and one chromosome goes to each central lobe. So, as you can see, they're separating with the spindle fibers pulling them apart. Telophase, the fourth phase. Daughter chromatids reach opposite poles and begin to condense. So as you can see, they reach opposite poles, and as you can, Cytokinesis. So what cytokinesis basically is the cell that divides into two by contraction in the actin fibers, the cell of the actin fibers, and the cell splits and furrows, pinches off closing cells or from each other, and then the cell replication is completed, and then after the cytokinesis, the cell cycle repeats. So these are like two daughter cells. Okay. More additional information. This is like a drawing of prophase. So it's gonna. These are like the different what's in prophase. So like the astar, the nuclear envelope, which is the black part, the centrosomes, which is the green part, and then two sister chromatids. So like two of these green parts would be a sister chromatid. The centrioles, which is like that part of the spindle fibers, and then each meiotic spindle fiber, which are these red parts. The asterisk would be like the two X marks inside the cell. So what this is, the nuclear envelope. It is as known as a nuclear. It also is as known as a nuclear membrane. nucleotinosis and karyothelica. Sorry, I'm pronouncing this wrong. It's a phospholipid bilayer membrane which surrounds the genetic material in nucleosis and eukaryotic cells. Consists of two lipid bilayers. The inner it consists of two lipid bilayers. So the inner nuclear membrane and then the outer nuclear membrane. So like this would be inner and then this would be outer. Separates the nucleus from the cytoplasm in eukaryotic cells. The astar is a cell structure shaped like a star formed around each centrosome during mitosis. The centrosome is an organelle near the nucleus of a cell that contains the centrioles and from which the spindle fibers develop in cell division. And then the centrioles is organelle near the nucleus, occurring in pairs involved in the development of the spindle fibers in cell division. And then sister chromatid, it refers to either of the two identical copies chromosomes formed by the replication of the single chromosomes, both copies joined together by a centromere. Spindle fibers form a protein structure that divides the genetic material in a cell. The spindle fiber is necessary to equally divide the chromosomes in a potential cell into two daughter cells during both types of nuclear division, mitosis and meiosis. In mitosis, the spindle fiber is called the meiotic spindle. I'm going like over my time, but at least you're getting this information. Last but not least, the second of more information. At the end of mitosis, the cell has two daughter cells. So, for example, back on this picture, two diploid cells, two daughter cells. It has one cell division, mitosis. Daughter cells are identical. The daughter cells are diploid. The daughter and parent cells are diploid. How many chromosomes? Okay, question. How many chromosomes are in the human cells? 46. A human cell has 46 chromosomes. This splits up 23 eggs, 23 sperm. This is one set of a chromosome, but both has 23 pairs. All other cells, like the liver, cardiac, blood cells, has a total of 46, two sets of chromosomes. So, two sets of like the egg and the sperm and the The diploid twin is two sets of chromosomes, which is equals 46. N is a unique chromosome, haploid number, which is basically N is a haploid. So within that you have one haploid. 2N equals 46 total. And that's all what I have, I think. That's all what I have for you today. So basically I taught you about chapter 12, mitosis. And I explained to you about what mitosis is, what the cell cycle is, what interphase is. Two kinds of cell cycles, which is the stomatic and the gametes cells. Cellular replication, and why it's so tight. And then interphase again, and stages of mitosis, which has 4 stages: prophase, let me make sure I get that right. Prophase, metaphase, anaphase, and telophase. Did I get that right? Yes, I got that right. And then more additional information that has to do with everything. So, I hope this was thoughtful, and I hope that I taught you something. Thank you for watching.